Well, thank you very much. I'm Tony Seibert, a retired professor of radiology at the University of California, Davis. I have the distinct privilege and honor of substituting for Dr. Mary Ellen Geiger, who is the principal investigator of the Medical Imaging and Data Resource Center. And I have also the distinct privilege of introducing Dr. Paul Kinahan, who is a professor of radiology and bioengineering and the vice chair of radiology research at the University of Washington in Seattle. He is also the head of imaging research laboratory and clinical director of the PET-CT imaging physics. His research includes optimizing physics of PET and CT imaging, the use of image reconstruction methods, objective assessment of image quality, and the use of quantitative analysis in imaging-based clinical trials. He was a member of the team that developed the first PET-CT scanner prototype some time ago, which was a Time magazine invention of the year 2000. He was a founding member and chair of the NCI Quantitative Imaging Network and a founding member of the Quantitative Imaging Biomarkers Alliance. More recently, he is the director of the Imaging Core Lab for the ECOG Akron Cooperative Group. He is a co-PI of MIDRC, the Medical Imaging and Data Resource Center, along with Mary Ellen Geiger. And this is a multi-society and multi-center initiative supported by the NIH and NIBIB for the rapid and flexible collection, artificial intelligence analysis, and dissemination of COVID medical imaging and associated data. I'm very pleased to introduce Paul and his topic, Curating the MIDRC Collection of DICOM Images for Use in AI Research. Paul, thank you for delivering this lecture today.
Thank you very much, Tony. I really appreciate the kind words and filling in for Mary Ellen and helping me get started here. Where did this go? Can you see the slides okay?
Yes, they look great.
Okay, thanks. Okay, so thanks. Yes, and my research interests now include informatics. I'm following in Tony's footsteps through SIM and informatics, it seems. I'm going to talk mostly about the curation aspects that we're going through in the DICOM images and hope to give an overview of that. But first, I just want to acknowledge that there's many people, including Tony, and Emily and Mary Ellen, Katie Pizer, the group that's been working on this. This is a large consensus effort, as you'll see, that we are not trying to reinvent the wheel. We want to work with existing standards in a measured and sustainable manner. And so it involved a lot of discussions, all of which are necessary, we'll talk about. I also want to acknowledge the support of the NIBIB contract that supports MIDRC, as well as the active engagement of the NIBIB staff and leadership in supporting the efforts as well. Many valuable contributions have come from them. So the next few slides will probably be a well known to everybody, but for those of you that have seen these before, forgive me, but just in case somebody hadn't seen these before, I want to give at least the brief context here. So I have about 4 slides of this. The first one is just, of course, the pandemic, and we were all scrambling to figure out what we could be doing here to help out. And one of the things, of course, that received a lot of attention, as we all know, or as most people know, was imaging using either X-ray or CT. Now, whether or not it has a clinical role is a separate issue, but there was certainly a lot of effort that went into using artificial intelligence or AI methods to facilitate any clinical work. And this led to the formation of MIDRC, the Medical Imaging and Data Resource Center. We have 2 PIs each from the American Association of Physicists in Medicine, or the AAPM, also the American College of Radiology, the ACR, and the Radiological Society of North America. So 6 PIs. And in addition, the site, because there has to be a single prime site, that's the University of Chicago, where Mary Ellen Geiger is the contact PI, and ably aided by Katie Pizer and her team there as well. So the goal of MIDRC was to provide a curated image and data commons. The idea is to enable researchers to address topics no individual archive could yield independently. And there were multiple image archives produced, both prior to the pandemic for other reasons, and during the pandemic for people trying to be helpful to provide data for algorithm development. The two main components within MIDRC are the data commons, which I'll be talking about. Then there's a fair bit of effort separately that you've heard about in other seminars on the machine intelligence and computational capabilities, and the really exciting work that was presented by the group working on radiology reports de-identification and as well as synthetic image generation in the virtual trials. But I won't be covering any of that here. So just in case somebody had missed it, it was pretty interesting. About a year after the pandemic, there had been literally hundreds and perhaps thousands of publications using AI tools on radiology imaging. And then the essential meta-analyses came along. And essentially, the titles of just two of the more influential papers, meta-analyses at that point, came out, which really point out that none of the algorithms were working or providing useful results that were translatable. There were many other, well, by many, I mean probably at least another 4 high-level reviews that all came to the same conclusion. And it was for a combination of reasons. The one that we're going to focus on here is the poor quality of data. And one of the papers coined the term Frankenstein data sets. Now, that's a specific case where if somebody well-intentioned is building a cohort of data and they include somebody else's cohort without realizing that they're duplicating cohorts because the other data they had has already been incorporated. And as well, so it just is the tip of the iceberg in terms of understanding the data. So essentially, curation would be the ability to tell you something about the data and the quality. Doesn't necessarily mean that you have good quality data in the sense of like the best possible images, but rather that you can say something about the quality of the images. It's good quality, bad quality, making sure it's not duplicated, that it's as much as feasible, it's traceable and has quality control, and as much as possible has external validation. And we'll come to some of these issues again in a few minutes. So the way MIDRC has evolved is there's a couple. This is another high-level diagram. From many imaging centers, we're collecting CT and X-ray images, radiographs, either CR or DR. And in addition, now specifically targeting MR as well too, which I won't talk more about here. And as of the end of last month, we now have almost 105,000 exams that have been released that are available to the public for free. Basically, go in and download everything and try to develop methods that are going to generate useful information from the images. There's a lot that has gone into this that you've heard about in other sessions, and I won't go into that. Here, I'm going to focus just on the aspects of curation that relate to the cohort selection. So, and by that, I mean, if there's 105,000 images and you want to start testing things, how do you pick out the images that you want to start working with and whatever the algorithm it is that you're developing? So this will lead us into some of the unique attributes of MIDRC, which relates to the curation harmonization, and something in particular which was new to me just over a year ago, but has now become entrenched in our process, something called LOINC, and I'll go into that in some detail in a bit. So to motivate the need for curation, here's two examples. On the left is from the early days when we started. This was from a collection of X-ray images of the chest, and so radiographs. And you can see by just at a quick look that there's a lot of variability in the quality, but also in what's in the images as well too. Some are AP, some are lateral. There's one in the center, which is actually a knee, a pair of knees, which is, even though the image was labeled as a chest image. So to give a sense for the variability there. These are in the images itself. On the right-hand side, we're now looking at the DICOM metadata for some CT exams. So this was a set of 5,500 CT exams that were labeled as abdominal CT scans. From that list of 5,500, there were over 350 different study descriptions, and you can see them listed here under study descriptions, as well as the percent of the total. So you can see that there's a long-tail distribution in the study descriptions that are being used here. These are just listed in the order of frequency. Oh, sorry, somehow that went the wrong way there. Okay. So both in terms of what's in the images themselves and also what's in the study description itself, there's a lot of room for improvement if you're trying to figure out how to select images to use for algorithm development or testing. So we were faced with a question like, what do we do? So there was two parts to this, and I'm going to talk about in more detail about the second part, but briefly, the first part is, well, what do we know about the images or the pixel data itself? And as you may know, there's been multiple methods to try to analyze images using AI methods to say something about the quality or what body parts are in the image. And there's been some success there. There's even more recently some newer methods being developed that are combined methods that combine multiple AI models that seem to have promise. What we don't have yet is a framework that allows these all to work together, and this is part of another MIDRC project that's underway that we can hopefully talk about at a future seminar. The second part we'll focus on here is about the free text fields in the DICOM headers, in particular the study description. So it would be great if we could have some sort of structured method, and I'll talk a little bit about RADLEX, developed by the Radiology Informatics Council within the RSNA. The challenge with RADLEX, although it is a well-structured lexicon, it has over 75,000 terms. And so it's not really well matched to the task at hand. We could, in principle, use a different standardized lexicon, but the consensus of the group working on it is that we really should not be creating our own descriptions. We really want to have something that's standardized and maintainable. We also, a challenge that we faced was clinically, there's a lot of value in having very precise definitions of the studies so that when a radiologist or other reader is reviewing the image, they know as best as possible the clinical intent and the scan protocol that was collected. On the other hand, the more detail, the more granular it is, the harder it is to generalize to the level of, say, for example, I would like to collect all the chest CT exams where there's contrast, for example. That'd be a more general description, which would be probably of more use to somebody who just wants to build a cohort of images to work with and to trust that that description captured the images appropriately. And finally, the approach we'd like to take was a fair one, which is findable accessible, interoperable, and reproducible, which is now becoming the standard for open data or open science even. So just a bit more detail about the DICOM format. The one particular element is group 8, or the tag is group 8 element 1030 is the study description. This is institution generated locally. Sometimes it's standardized, sometimes it's not. Even if it is standardized, it can be modified on the fly to try to collect variants. And sometimes it just has mistakes as well, too. For example, the body region may be incorrectly identified. As those of you that have worked in imaging centers know, the technologists at the time of acquisition are trying to capture the relevant information for the clinical purposes, which may not which clinically may work quite well locally, but it doesn't make it easy to pull the information together across multiple sites in a harmonized fashion. A couple notes I want to add is that one thing is that MIDRC does not modify the DICOM image. And for example, we don't take the study description and put our own in. We try to touch the, other than removing PHI, that may persist, we try to not touch the images at all to preserve as much information as is there. But so we were looking for a standard, and one of the things that came up, which I'll talk about in some more detail now. Sorry, a plane just flew overhead. We'll talk about in more detail is something called the RADLEX. Oh, I talked about the RADLEX effort. The RADLEX group also went a step further and developed something they called the RADLEX playbook, which I'll talk a bit more about. But this was really a set of study descriptions with in a structured manner, with a whole bunch of secondary information such as presence or absence of contrast, timing information, things like that. And this was being developed by the RSNA informatics group, as I mentioned. Interestingly and separately, a group called Regenstrief, an institute, was taking something called the LOINC codes, which are being used in lab medicine and other branches of hospital diagnostics. To Regenstrief was looking to expand LOINC, which I'll talk about more, to include radiology codes as well. And fortunately, the two groups met up. I actually don't know the history of how they became aware of each other, but it's not that surprising. And they agreed to merge efforts, and they created something called the LOINC RSNA Radiology Playbook, where they moved over a large component, perhaps all, I'm not sure, of the RSNA RADLEX playbook into LOINC. I should note that the playbook, the RADLEX playbook, is still available, but it's no longer being developed. All those efforts have shifted over to the LOINC RSNA radiology playbook, which I'll talk about now. Oops, I went too far there. So LOINC itself, that's short for logical observation, identifiers, names, and codes. This was developed in 1994. But the a group called the Regenstrief Institute and this is what was been expanded to include the RADLEX playbook or core playbook. LOINC is part of the UMLS, UCUM, and other standards I'll talk about, and it's free. I encourage you to just go to the website, just Google LOINC, LOINC, and it's free for anybody to download or interact with. It's quite a powerful database. And but to back up a second before that, because as I mentioned, one of our goals was not to invent a new standard, but rather adopt one that was sustainable. And LOINC meets that because it's part of the are recognized by something called the Unified Medical Language System. And this is maintained by the National Library of Medicine. And essentially, it does a lot of things, but the part that we're most interested in is at its core is something called the metathesaurus. And it tracks a large number, about 100, of controlled vocabularies and classification systems. So these would be things that many are familiar with, such as CPT codes, ICD-10 codes, mesh codes, SNOMED CT, Loink, and others. Actually, and I should mention that for those of you familiar with the SNOMED CT, there's an agreement was reached between SNOMED CT to start looking at establishing correspondence between SNOMED CT and Loink codes, which will be something to look forward to. I think it's early days in that effort. So just to establish the credibility of Loink and the sustainable nature, it's been in operation for some time, as I mentioned. And right now, actually, I just checked and it has over 100,000 codes. Now, these would be things for like blood panel tests, serums, you name it. It's well structured, and it's incorporated into the HL7 framework. So it allows for both the ordering and reporting of diagnostic tests through the hospital networks. As I mentioned, there was this combination with the RSNA efforts. The subset that's specific to radiology has just over 6,700 radiology specific Loink codes. And I'll give a example of that here. So if you just go to Loink, as I said, anybody can do that. There is a syntax for accessing the database, or you can just download the entire set of CSV files as well too. But they have a pretty powerful interface. So in this one here, I specified, if you see up at the top, it says class rad. So that means all the radiology related procedures is a class. And method is specified as CT, and I'll come back to that. But essentially it says the modality is all the CT, and the system is chest. And what that means is the part of the body, which is the Loink terminology for the body region, is chest. With that, it filters down to 217 Loink codes. And so I'll just take the first one as an example, if you can read that. So there's a unique number. There's no significance to the number other than the fact that it's guaranteed to be unique. There's an associated long common name, which was the particular payload of interest for the MIDRC group. The long common name is guaranteed to be unique, and it's actually comprised or generated using an algorithm that guarantees uniqueness and pulls together all of the ancillary information about the scan. So this first one that we're looking at says CT chest 3D post-processing without contrast. And so that's actually formed by an algorithm that Loink uses with the RadLex team to make that a guaranteed unique name. And it looks an awful lot like what we would in radiology say is a standard study description. As a matter of fact, in some cases, they're indistinguishable. One quirk that almost always gives it away, though, is if you look at the bottom row, you'll see CT chest without and with contrast IV. Intuitively and typically, in radiology departments, it just seems that we always say with and without. But there was a good reason, which I've forgotten, that the Loink RadLex group agreed on having without and with. So that's one little thing that shows that this is a Loink code and not what somebody typically would enter into a center. So we're looking here, though. I've only covered two fields, the Loink code, the number, and the long common name, and you can see columns for other components in the Loink codes, and I'll talk a bit more about that. The Loink structure has the code, the fully specified name I mentioned. It's actually not, there's a nuance there, but I won't go into that. essentially there's 44 elements associated with each LOINC record. A lot of these are not relevant to radiology. They're for different aspects of, say, lab medicine tests or things like that. But as I mentioned, the great thing is the key thing is the long common name, which is checked for uniqueness. And the other thing to sustainability is this file, the whole LOINC database is reviewed and updated 2 times per year. People can suggest new codes. So if you're for some reason looking for a code, and it does happen that there isn't one that describes things precisely like you'd would want, you can propose it and they will review it. And if it fits the system and it's properly justified, it'll get added to the database. So here's the 44 elements, just so you can see. I won't spend any time on this, but there's some of these are more relevant than others. And these are the ones that are in the main database. There's also secondary databases that we can go into if there's interest in that. So as I mentioned before, I just established the correspondence between the DICOM metadata we're familiar with in LOINC codes. I've listed a few of the key things side by side. So the first one is the code itself for LOINC, and DICOM has no equivalent. There is no, we do have UIDs in DICOM, but there isn't anything that says defines a unique protocol in that regard. DICOM has a study description. However, as I mentioned, it's variable. It depends on what the operator types in or what post-processing steps how they will modify the study description. The LOINC code here, though, as I can't emphasize enough the value, it's unique and it's generated by an algorithm. So it's a very precisely defined term. And then the other things that matter are in DICOM, we would talk about modalities such as CT, DR, CR, MR, things like that. In LOINC, it's called a method, and I'm assuming that's because that term had already been set up in the LOINC terminology. Similarly, what we would call body region in DICOM is called the system. I should also mention that for both the method and system, modifiers are allowed. So you could have XR for X-ray as a modality or method, but you can also append portable to that. So if it was a portable X-ray system, you can note that in LOINC, and it's again a controlled vocabulary. And last is one particular interest to us, of course, is contrast. And contrast, the presence of contrast is not in the LOINC record by itself, except in the long common name. However, this information is stored in one of the secondary files, and that information is pulled into building the long common name, so it's obvious if contrast is there or not. So with that in hand, the core of the approach that we've taken is illustrated here in this table. So we have something that we call the MIDRC LOINC mapping table, and in its current form, we've tried a couple variants earlier on, but in its current form, it takes in the DICOM metadata on the left, and the first column is the modality. The second column is the study description. The column in between is just the frequency in this particular analysis. And then that gets mapped to a LOINC code, which has a one-to-one correspondence with a long common name, as you can see some examples. And then associated with that are some attributes, such as the system, which is the CT, the method, which is CT, the system, which is chest, and other things that are important that we'll come back to. But this is really the core of the process that we're using right now. You can see a couple modifiers there, I think, if you can zoom in at all, but under the column, the second to the right column, under method is, you can see ct.angio. So the angio is an allowable modifier to say that it's being used for angiography. Similarly, in the system, you can see we have chest, and we can have abdomen and pelvis, and there's other variations to indicate a specific area of interest. Anyways, let me just very briefly talk about the process. So this is you may have seen the versions of this diagram. This is the data flow through MIDRC, starting at the contributing sites all the way to the external users. I'm going to focus just in on this process where we're creating and using this mapping table here. So we start with the DICOM images coming in. We abstract them, or sorry, the ACR RSNA teams abstract them, and they provide the manifest that describes the data to the Gen 3 group, which then makes it available in the Data Explorer for people to select images. In parallel, the abstractions are also providing us the information that we need, the study description and frequency as well. And then from that, the data quality and harmonization group creates both the mapping table and something called the attributes table. So just to give a kind of an algorithmic overview of the process, this would be the information that comes from ACR and RSNA. So for example, the modality would be DX. Study description is XR, chest, OneView, AP, and this one is the most frequent one. So we have a process that has evolved as we're now approaching our third version of the table. And I won't go with this into any detail, but essentially we go through a series of updating it on a regular basis. The other thing is there's so many study descriptions and it's such a long-tailed thing. We don't attempt to map all of the studies. We only work on the ones where we have at least 10 instances of a study description, or 10 or more. And if that occurs, we'll map it just to because of the feasibility of the time required. The last part comes from the LOINC codes. We also have the attributes, and so we could talk about whether on the left we have a series of LOINC codes. Just here's 100, for example. Then we can sort them by, or we can filter out just the CT studies, and then finally we can filter out the ones that are CT and with contrast in the chest. And so that's the way the attributes is a separate table that allows us to do filtering for the cohort selection, this is the Data Explorer. And so if you can see it, it's a little hard to see on the PowerPoint slides. But I encourage you to explore it. Just go to MIDRC and you can launch this and see the studies. And now with the combination of the attributes and the mapping table, you can now go through and sort by the LOINC properties, which would be the long common name, or it could be the method, which is the modality, or the system, which is the body region or other parts. My last slide here is I just mentioned that we're addressing the need for a large, freely available and curated imaging data set. And although DICOM was a great start, it wasn't quite sufficient. So we've added in this extra step here to help harmonize the study descriptions using the LOINC terms as the first step. And so far, in our earlier evaluations, it seems to be working quite well, where we can map over 95% of the studies or exam descriptions to only 65 unique LOINC terms. In other words, we're keeping it fairly coarsely grained so that you don't have to know too much detail about what it is that you're selecting. And then, as I mentioned, we're expanding this to MR and looking for alternative methods to try to address the long tail situation. So I'll stop there, and I really appreciate everybody's attention, and I'll hand it back to Tony.
Well, thank you, Paul. I've been part of the group, and this is a monumental effort for sure. And Paul has really done a fantastic job. A pat on the back to Paul for being able to head up the data quality and harmonization subgroup of this really very, very large effort. Now, certainly, if anybody has any questions, feel free to put them in the chat. I have a couple of questions as well. And one of them was already answered. It was, what role did the FDA play in the MIDRC initiative? and I believe Emily answered that. And you, did you want to say anything about that, Paul? The FDA,
Yeah, I can
did they-
add a bit.
Yeah.
I think specifically for this part here, the data quality and harmonization, the FDA, sorry, let me back up one step. So several people from the FDA have been involved with MIDRC since the beginning, from the creation. And MIDRC covers a lot of territory, while the FDA people that are, to my knowledge, none are currently on the data quality and harmonization committee, but they're certainly aware of it. Their efforts have been going into things like data sequestration, which is a critical thing. It was on one of the slides I skipped over, but 20% of the images have been set aside, as well as developing methods for, if you're going to test an algorithm, what are the metrics you should be using? And the viewpoint is that later, if somebody is going to be using MIDRC to test algorithms and potentially taking it to the FDA, they're being given some guidance about what are the appropriate tests and what data they should be using and things like that.
Well, thank you, Paul. One of the questions that, being on the committee, has the MIDRC DQH group proposed any new light codes? Or would we do we wish we had some different codes than what's available?
Yeah, right. I think we do. So I mentioned that to date, no, but there was a couple times we were getting pretty close to it. But then we discovered there was something that worked quite well. We, in addition to the two main tables, which I forgot to mention, by the way, they're on GitHub. And the process, it was in that table, I think, that I went through a little quickly. But so the abstracted information about the CT data or the modality and site description is uploaded by RSNA and ACR to GitHub. We take that and create the updated MIDRC table, which is free to download separately from those two main tables, or the attributes table is the other one. Is we have a kind of a handy list. It's our working list of things that we've used repeatedly, sort of ordered by body part or body region, and modality. And so far, it's turned out we haven't needed any. But it's surprising, even with this relatively short list, how many combinations that we need, such as abdomen, pelvis with contrast. Could be something that somewhere has just never come up, but we haven't hit that one yet. But we're ready. We also had a preliminary discussion with the Regenstrief group to say, look, if we find something, can we propose it? And they're welcoming our participation.
Well, before I get to the other questions, I have one more question that I want to ask with respect to the DQH group. How is it that, do you have experts on the group that help assign these codes to these images? Because sometimes the study descriptions are a little less obvious.
Right. Thanks. Yeah. Well, we have this one famous expert, Tony Seibert, the RSNA gold medal winner. But no, we have radiologists and very experienced people working with protocols. And then if there's a question, we can go and spot check images. The Data Explorer now provides very nice tools with using the open source OHIF. OHIF is the acronym. It's a very, very, very useful DICOM viewer. And so we can take a quick look. We can't review every image. It's just, there's 100,000 exams, each exam containing multiple series, and some series containing hundreds of images. So it's not possible to check them all, but we do some spot checking. It does point to the need, though, for probably, literally or ironically AI-based methods to help curate the data at some point as well.
Well, we do have a really broad-based attendee list here. We have one question. It's called, do you have any gynecologic oncology images and data? Can this be outsourced for collaboration?
We almost certainly do somewhere in there. Partly because although we asked for chest and chest X-rays and CTs, we got everything as you know, and so there's ultrasound images, there's PET images of all and it's across, and there were MRI, you name. We even got some bronchoscopies, and so probably somewhere in there are gynecologic images. To find those might be a little challenging right now, but it's not out of the question. It might take some digging. Later, as we expand the curation efforts, we hope to be able to identify everything.
Yeah. Well, thanks, Paul. And that begs a question before I get on to another question. You have this long tail issue, and you cut off the number of instances of a specific type of study description to 10 entities. So can you still get access to those images that are in the long tail? Is there a way?
Yeah. Thanks for saying that. No, so all the images are there. It's just that we've only been able to map about 95% of them. So, but using LOINC codes. If you don't use the LOINC codes, you can just go by the original study description. They're still findable if you know what to look for. And then, of course, the other thing is once you download the images, you can always look to see what's there as well too. The challenge is that in that case is you have to download them first to see what's there.
Right, and for those studies that don't have a study description, they're in that long tail.
Yes.
How are they addressed? Because let me just tell you, in developing a data set that we want to send from UC Davis to MIDRC, there's a lot of studies that for some reason do not have an associated study description because it's not necessarily required in DICOM.
Part of it is, I think, due to the fact that, which I didn't talk about much, is the images are de-identified before they leave the site, and there's a couple of recommended processes by the ACR and the RSNA, but the sites will do what they can, and we're grateful for the images that we do get, but there have been times where literally everything in the DICOM header has been wiped out. We even have to guess at the modality in some cases. So for some of those, there is a code called unspecified body region, so we can use that. Perhaps amusingly, the original or the current LOINC system description for unidentified body regions is XXX, the three letters. And so we decided we probably shouldn't use that. So we changed it we created a new term just called unspecified. So those are kind of our catch-all. Some cases we can go through and look at the images directly if we have time, and we could identify them. The concern is that when new ones come in without a study description, which they will, we don't know anything about those till we look at them. So we took a vote of the committee after some discussion and decided that those are always going to remain just unspecified. And there's not really a good workaround. Fortunately, it's a very small fraction of the overall collection.
Okay, another question. How do you handle it should a commercial entity show interest in the data set, the MIDRC data sets?
Well, the MIDRC data set, to my knowledge, is free for anybody to download. If you want to use it for commercial purposes, there's some information on the website about what's available. I haven't been keeping up with where that is right now. But we have had people download the entire data set already. It's open. You just register and download it. I would add that, the ideal, one of the ideal outcomes of MIDRC is if somebody uses it to develop an algorithm that's effective and or somebody uses that to develop a clinical product that's effective and shows that either develops it or validates it against the MIDRC data. That would be a wonderful accomplishment if we're using this, because I think many of us realize we need industry for sustainable clinical solutions. I mean, the research that a lot of us do is great, but it's not going to get directly to patients unless industry is involved. So industry has always been a key part of this from the beginning. Where it is in terms of licensing or FDA claims, I'm not quite sure yet though, but that should be on the website.
Thank you, Paul. Another question: at what stage is the PHI de-identified in the workflow?
That's a good one, and it was in one of the diagrams I flew across, but actually it's at multiple stages. So first, when it leaves the site, by whatever means that their IRB or their local team is using, and then it comes in through either the RSNA and the ACR. And both the RSNA and ACR have technology stacks that use both automated and human reviewers to look at images to be sure that it's properly de-identified. And there's some evaluation about they're not using methods that are roughly the same and roughly the same profiles for the metadata de-identification, but there's also some interest in using automated methods as well too. So the images are generally scrubbed first by the submitting site, they're reviewed, and then if necessary removed by the MIDRC groups. Typically we don't alter the information though. I think sometimes the data might just be deleted if it contains too much PHI. I'm not quite sure where we are in that, but it does point to a real tension in the research. Because on one hand, you want to protect the patient confidentiality, but the best way to do that would be to not provide the image at all. Then there's no chance. So if we're providing an image, how much de-identification still leaves useful information? And that's a navigation that's still going on in practice. And I know that there's groups at the cancer imaging program associated with the Cancer Imaging Archive that are trying to come up with recommendations about how to adjust the dial between enough de-identification to protect patient rights, but not so much that it makes the data useless. And that's still not a solved issue to my knowledge.
That's a really key point. Another question, perhaps the penultimate question, because I have one more question too. Do you have tomosynthesis images and reports?
We don't have reports for anything. I'm pretty sure we have tomosynthesis images by now, but it would be down in the tails. We have a lot of portable X-ray images, and tomosynthesis is one of the modalities that we do categorize. We'd actually have to take a quick check. I don't think there's a lot. But there could be some. Tony, do you remember seeing any? I don't recall.
I don't recall seeing any at this point in time, but I'm sure there's some in there, because stuff. I mean, I'm trying to send images, and a lot of stuff leaks through, and I'm sure there are some. But I think with long COVID, that's another thing that I think MIDRC is shifting towards. And maybe you can comment about the long COVID issue and
yeah,
what is MIDRC planning on doing with respect to that, and with respect to capturing images.
Yeah, thanks. That's a good one. Thanks. There's as many people are aware, there's an increasing number of papers saying that long COVID is really quite a serious issue, and it's going to be with us. Some people who have had acute cases of COVID, some large fraction, and the most recent paper, I think, said 39% will have persistent symptoms for undisclosed number of years, because it's all new. We don't know. But this is affecting multiple regions in the body. The one recent paper using MRI found changes in all sorts of organs, and some of them are asymptomatic, some are symptomatic. So but definitely areas of cardiovascular system, neurological issues. So one of the things MIDRC is deliberately including is things that were the early indicators or concerns. So we're looking at MRI specifically, and with the intent on neuro images and cardiac images as well too, to start getting that up to speed. And so we're just expanding the data dictionary to include the key terms for MRI data acquisition right now.
Well, thank you, Paul. You know, one final question. I know that I'm part of the data quality and harmonization group. What about data quality? Just a really brief, what we're planning on doing with respect to data quality.
Yeah, so we at least my recollection, curious to see what yours is that we started off as a bunch of medical physicists saying, oh, we're all over the data quality stuff. And we talked about having a MIDRC data quality certification and things like that. And we ran smack dab into the, well, how do we find the studies if we can't read the study description? How can and so we had to pivot to this metadata on the informatics side, which is why I was kind of kidding. I'm trying to copy your career now and learn at least a little bit about informatics. So it's still on the list and it's still really important. But I think what we've done so far is help set the framework. Because to have information collected about the data quality, is it a noisy image? Is it low resolution? Are there artifacts? Those sort of things. We need to have a way to track that information. And I do believe that what we've built here is in particular with the Gen 3 group at the University of Chicago, who has a data model, is a way that we can also now start looking at how do we track that information and including it in the data model as well. And so that's never gone away. It just had to take second seat to the primary goal of letting people download images.
Well, thank you, Paul. You can see that there is a very, very long-term commitment to this effort, and it's just expanding, which I think is very exciting. And we are really very, very happy to have you give this talk today. I would certainly like to appreciate everybody's attendance. Thanks for joining. This has really been, to my mind, something that really is necessary for the word to get out on, because it's such a valuable resource and it's going to be expanding, and being able to provide the medical physics component of this is really an important aspect that is really going to make this database that much better. So thank you all for joining us. Thank you, Paul, for giving this excellent talk, and we look forward to future communications on this effort.
Great.
So it's been great.
Thank you, Tony. Much appreciated.
Sure. And with that, I will close the meeting. Thanks all for attending.
Sure.